So welcome back. Thank you everybody for sticking with the workshop for the last 2 weeks. We really appreciate all of the time and attention, and we are very excited to work with you over the course of this weekend on building some CompuCell models. So this is officially day one of the hackathon. This session is being recorded and live streamed to YouTube. By this session, I mean my intro material this morning. We will stop the live stream once I finish presenting the intro material so that people can speak more freely about their projects during the lightning talks, but we do have some preamble to get through. So slide decks and materials are available in the student materials folder as usual. Starting today, we will be populating the hackathon folder, more so than the student materials, sorry. Everything for last week and the week before was in the module slide subdirectory in the student materials folder. We'll be putting what we do this weekend in the hackathon folder. Our teaming doc is up as well. If you have not looked at that yet, now is certainly the time. If you have a project idea and you have not added it to the teaming document, please go ahead and do so this morning. And then people can reference that throughout the day today to help orient people towards potential project groups. Just like to go ahead and thank our funding sources through the NSF and previously through the NIH, as well as our ongoing support networks through the IUB Biocomplexity Institute, as well as the OpenVT and Glimprint initiative. Okay, so just talk about a little bit about the schedule for this weekend. So for today, we're going to be mostly focusing on teaming and helping people find groups and initial project setup. Day 2 and day 3 will be predominantly work time with your groups, with instructors and helpers kind of circulating around to assist with your models. We'll do another round of lightning talks this morning until about 11 o'clock, maybe a little bit later because we're getting a slightly late start now, but from 11 to 12, we'll have kind of an open teaming session where people can move around in breakout rooms and discuss projects, figure out which group they might want to join if you still have questions. From 1 to 3, we'll do project setup. We'll do a session of project setup, scoping, and planning. We do recommend that you spend at least a couple of hours this afternoon, working on some goal setting, making sure everybody in your project group is on the same page about what you're trying to accomplish by the end of the weekend. From 3 to 4, we'll have a group check-in, so everybody can let us know how things are going, what they feel like they might need over the course of the next 2 days. And then from 4 to 6, you'll have 2 hours of work time with instructor help. And we usually stick around a little bit longer than that, depending on how many people still need immediate help. But 6 p.m. is kind of our official cutoff time for when the instructors will probably need to go and get some dinner. And then we will meet back here tomorrow at 10 a.m. again. We'll do a quick morning check-in to see how things are going. You can let us know if there's been any updates over the evening. We know some people in different time zones work through the night, so you can let us know in the morning what help you need. Then from 10:30 to 1, you'll have a morning work session. From 1:30 to 2, we'll do another quick afternoon check-in. Again, these are just to find out how things are going and what people need. These are not formal check-ins, so don't worry about where your model is or what you still need to get done at that point. From 2 to 5, we'll have an afternoon work session, and then we'll do first day reports in the evening before we sign off for the night. Day 3 will be very similar. You'll have slightly longer work breaks. We'll have morning check-in, work block from 10:30 to 2. From 2 to 2:30, we'll do a quick check-in. 2:30 to 5, you'll have to work on models again. And then from 5 to 6, we'll have a slightly more formal final report session. We'll send out a slide template tomorrow for you to start filling in. Just a few slides on reminding us what your project is, what your goals were, challenges you faced and how you overcame them if there were any major ones, and then just a quick demonstration of where your model prototype currently is at the end of the weekend. So some notes on logistics. I know most of you saw this yesterday, so I will try to hurry through this as much as possible. Just a reminder though, the main room that we're in now will stay open. We'll use the main room for daily check-ins. So we'll have everybody come back to the main room for those half-hour check-in sessions, and then you can return to your breakout rooms to continue working after that. And this main room will stay open past the end of the day. So we technically, the number of the instructors will leave at 6, but this meeting room will stay open through the night. It usually times out somewhere around between midnight and 2 to 3 a.m. Zoom has a 14-hour limit on how long meetings can go. Just some reminders on our people. We've gone over this a lot, so I think we'll just be quick today. Today we currently have in the room me, Dr. Glazier, Gilberto Thomas, Jill Vannon, who is another member of the CompuCell lab at IU Bloomington, Pedro, who you all know quite well at this point, and Yi Jiang, who I believe was a previous student of James. So they will be assisting us today. We may have some other instructors. Yes, Georgia State University. Thank you, James. Sorry. We will have some other instructors popping in and out throughout the course of the day, and we will let you know when they pop in. If you have specific requests for people, we can send them to your group. Gone over this yesterday. We will share our model building template when I'm finished here. And you can use that to kind of think about how to structure your model today in the afternoon, if you like. You don't have to use our template. It's just one suggestion. Okay, so I think I'm going to go through, we'll come back around to our lightning talks, but I want to give a quick overview of who is presenting today. So we have 2 new talks today. We've got Loïc Comeliau and Micah McKanstry. Loïc and Micah, if you want to send me the title of your talk, I can put them in the slides before we get started here in a minute. We would also like people who already presented last week to do just a brief representation of their slides, if that's okay, so that we can kind of get a refresher on what people are wanting you to do over the course of the hackathon. So we'll have Anastasia, Nasheen, Dipanjan, and Clara, and Mark represent again, if you don't mind, just for a couple of minutes. Okay, so some notes on project planning. I know we discussed this yesterday, so I'll do kind of a brief summary of it now. We'll do another extended version of this, maybe in the afternoon, for people who want to come talk a little bit more about the details of all this. So as many of you remember, we talked last week during our fundamentals of modeling session about the 4 different elements of computational models, particularly biological models. This idea of things being broken down into objects, behaviors, interactions, initial and boundary conditions, and dynamics and events. The key thrust here being that we don't get any of these things for free in our models. We must specify basically everything that goes into a model. So what is represented within a model is completely up to us as the modelers. So there are some questions we can ask ourselves before we get started. Again, we'll skip the activity portion of this so we can get to lightning talks. But as people give their lightning talks, and as you're thinking about which groups to join, some questions you can ask yourself about how to sort of set up your initial model prototypes is, what are the specific hypotheses you wish to test? So first of all, categorize your, or not categorize, but describe your biological problem of interest. And then think about what exactly you're interested in looking more closely at using your computational model. What about the system are you trying to demonstrate with your model, or at least with your model prototype at the end of the day? How can these things be measured? Can they be measured in the real world? And how does your model correspond to how those things will be measured experimentally? What are the key components you wish to model and why? So you're not going to be able to model every single thing about most biological systems because they're very complicated, but which things do you need to model in order to answer your questions posed in your hypotheses? And then, of course, what are the main processes, interactions, dynamics, and events you wish to model, and why are these necessary? That why are these necessary question is kind of an important component, because if you can't come up with a good answer for why it's necessary for this particular model, then the true answer is that it's probably not. So it's important when you start listing the things that you want to include in your model to ask, is this required to answer the questions that we're posing with our hypotheses, particularly in a hackathon setting, because you have limited time to prototype the initial versions of these models. So we also talked a little bit about the workflow of model development last week. This is a long, kind of intensive process that is targeted more so at the process of building models for publication and knowledge generation and sharing. So this can take a long time to really work through each of these steps systematically. You'll find during the hackathon with the limited time that a lot of your time will be focused on this final step of implementing choices as your simulation. So the point of this is to get practice in actually building a model using what you've learned for the past 2 weeks. But we do find that even in a hackathon setting, it's very important to still do some version of the first half of the modeling process as well. So some abbreviated form of looking at the system you want to model, defining your initial questions and hypotheses, thinking loosely at least about a conceptual model of how your model will function given its parameters and various things that need to be kept track of. Quantitative model template. A lot of cases people usually go find, so for example, if you're using a network model or if you need ODE modeling of a system, it's usually easier in a hackathon setting to find some pre-existing model for that, unless you are already an expert in writing ODEs. And then choosing the methodologies for the problem solution has already kind of been handled for us since we are modeling in CompuCell for this hackathon. But again, the point is you'll be mostly focused on implementation, but it is useful today to do some upfront thinking about how you're going to implement the model conceptually before you start coding. And again, to help you do that, here's some questions. What exactly are we trying to do with our model over the course of the weekend? Again, remember to always tack that onto the end of your thinking about the model. Obviously, you want it to be as accurate as possible, but that what are we trying to do over the course of this weekend is an important framework for thinking about your model for the hackathon so that you can kind of scope your project down into something that can be realistically prototyped over the next 2.5 days. So what questions is our model trying to answer? What hypotheses are we testing with those questions? What is the simulation's function? And then what do we want or expect the final results to look like? So this is also an important piece to do a little bit of prediction about what kind of output you're expecting from the model if it all goes perfectly. That way, when you do finally start getting some output from your model, you know whether or not it's behaving as expected. Another big question to ask before the weekend starts is, can the phenomenon we're modeling be broken down into component parts? So do we have to model this thing as one big chunk, or can we start with individual pieces of the system? How long will it take to model each part of the system? Another question to ask in this process is a single component enough for a 2-day project? And that goes along with which piece is most foundational, which components do we need first? So if one single component is enough for a 2-day project, then you'll have to identify which one is the most important as far as like kind of the foundation of your project idea. And then you can always add complexity later once you get that piece working, but it's important to identify which piece is most critical and determine if that's enough for a 2-day project. And if it is, to focus on that as your prototype, which you can then add complexity to later. So some general thoughts on team logistics. This is also important to consider in a hackathon setting. It's important to remember you will have 2 days to prototype your project. So again, it's almost every group we've had in the past leaves the hackathon with a functioning prototype of their project. It's not a full model of the system that they were interested in studying, but everybody usually is able to get some kind of prototype working. But it is important to remember that you'll have roughly 10.5 hours of free work time during the day, and this is with instructor help. So obviously people can continue to work offline over the course of the weekend, but the instructors will be available over the course of the weekend for about 10.5 hours. So it's important to think about what needs to get done while the instructors are still in the room um, during that time. Some important questions to address with your group before you get started is, what is the end goal for the weekend? What do we all want out of this by Sunday evening? What is each person going to do? Maybe talk a little bit about your backgrounds and how each person is interested in contributing to the group. How will the pieces of the model that various people are building be combined at the end of the weekend? So this is especially important if you have multiple people working on code, that those people are communicating so that when it comes time to take the various pieces of code and combine them into one simulation, that they work together as smoothly as possible. It's basically impossible to have the pieces come together completely smoothly the first time, but communication ahead of time can make the process of integrating them at the end much easier. The next question is, what don't we know? So in order to build our model, what do we still need to learn, and when are we going to learn it? So these questions are important because the question is, do we need to learn everything up front, like today, before we start coding, or is there something specific we need to learn, and then we can begin building our model and kind of pick up the other information as we go? And then what is the protocol when things don't work? We have very little drama with groups in these hackathons, but it is an intensive 3 days, and there are bound to be times here and there where there's miscommunications or things don't work as planned, and that can get a little frustrating sometimes. So it is useful to have some kind of agreed upon protocol on what to do when things aren't working, maybe how an agreed upon process for debugging, how to identify issues, things like that. If anybody is having any kind of issues with conflict resolution or things like that, you can always come to the main room and talk to me about it, and we will help you resolve that and get the project back on track. So if you're facing any kind of real roadblocks in the process, please let us know as soon as possible and we can get help out to you. So I think we will go ahead and start our lightning talks today with Micah.
Good morning, everyone. My name is Micah, and I want to present my project idea on modeling inflammatory bowel diseases that drive extracellular matrix breakdown in the GI tract. Inflammatory bowel diseases, including Crohn's disease and ulcerative colitis, involve chronic inflammation of the gastrointestinal tract. A key pathological feature is the degradation of the intestinal extracellular matrix, which provides structural support to the epithelial barrier. And according to the literature I've reviewed, this is largely driven by inflammatory cytokines such as TNF alpha and INF gamma, or tumor necrosis factor alpha, which stimulate the secretion of matrix metalloproteinases, or MMPs for short. Extracellular matrix loss comprises the cell anchoring, compromises cell anchoring, which weakens the tissue integrity and enables further immune infiltration. My motivation for this problem is to model how inflammatory cytokines drive extracellular matrix damage through MMP expression, and reducing structural support and promoting a feedback loop of like epithelial detachment and immune cell penetration. And I think that the applications project could be used to like model how we can develop medicines or drugs that may be able to like stop this loop or help treat this condition in patients. And the goal for my goals I want to hit when I begin modeling this is just like quantifying at what point this ECM breakdown is going to lead to like tissue destabilization, or demonstrate that there's like a correlation between the cytokine concentration and extracellular matrix loss. Or maybe looking at like what threshold does the extracellular matrix become irreversible or severe. And believe CompuCell has the necessary tool set to model the force stresses, decay, secretions, chemotaxis, everything that is necessary to begin like work on this type of project. For my initial inputs and outputs, I would start with a layer of epithelial cells, as well as maybe a few secretions from the epithelial layer, as well as like macrophages or anything else involved to kind of get everything started. And maybe like for the output, we could do a extracellular matrix integrity score, or epithelial adhesion stability. And this is just my literature I've reviewed on the slide right here. And before I conclude my lightning talk, I have beginner experience in modeling, which is mostly just in R programming. I've done weighted gene network code network expression analysis before on the left heart ventricle, where I analyzed what genes were highly correlated in side of the left heart ventricle. And I have extensive experience with Python, and I'm really open to taking anyone on my project that's interested.
Um, I was going to say that's a fascinating project. With some things that maybe weren't in the discussion sheet were how big a volume of tissue do you think you need to simulate at a given time? Maybe add these to your notes, Hayden, as well. And do you think you could do this in a 2D section, or do you need to do a full 3D model? Could you do a sagittal section of the tissue, or do you need to do the full 3D structure?
So I'm thinking about doing a 2D model and using the periodic boundaries on the x-axis to kind of simulate the tissue surrounding the area when I begin modeling it. It will be more of a cross section.
Right. Okay. And then I guess the other question is, are there any models now? Now this is gut epithelia, but not in a region where you have crypts, so it's not colon. Or is it, or is it a uniform epithelium? Is it a stratified epithelium? What kind of structure is it that you're trying to model?
For simplicity for now, I think I would just focus on uniform colon tissue.
Okay. Okay. Great. And I guess the only other question then is, are there any existing models that are relevant, published models that are relevant to this that you could look at that could give you ideas?
Not from what I've seen yet. I'm still looking just to like gather more information on what's a good template to base everything off of.
Great. Okay, perfect. Thank you.
So I already presented this talk before. So we're quantifying the dynamics of MTB, Mycobacterium tuberculosis-induced granuloma. So tuberculosis is a disease which caused by this bacteria, Mycobacterium tuberculosis. And so after the MTB infection, so after the MTB infection, the lungs, in the lungs, we will get a structure like granuloma. So what is granuloma? That is a cluster of immune cells where the bacteria and the infected macrophages are in the center, and there are other immune cells like T cells, dendritic cells. These are clustered around these infected macrophages to the release of cytokines and chemokines. So my goal is to I'm not going into the motivational part because we have also the experimental data, but I think in the hackathon I can't really, I mean, do the comparison with the experimental data, but I'll do a simple model. So I'll try to replicate the process of granuloma formation by just including the basic things like macrophages. And so initially we will include okay, so before going into that, what's our aim? First aim is to introduce the cellular POTS model, CompuCell3D, to generate the granuloma dynamics. Second point and third points are, I mean, it's not really possible to do in this hackathon. So the plan is that first, I'll put some bacteria and the resting macrophages in a 3D kind of thing. We can also confine to 2D if possible, and then model the interactions between them. So if the if this resting macrophage is near to the bacteria, it will catch the bacteria and become the infected macrophage. So now the infected macrophage could uh, do the secrete the TNF alpha or different cytokines like in interferon gamma, and it will attract the other cells like CD4 T cells, CD4 positive T cells. So these I will model here, and what my goal is to get a simple structure of granuloma where there will be infected bacteria and the infected macrophage in the center, and the other immune cells are surrounding it. Like for this hackathon, I'm just only considering the CD4 T cells, CD4 positive T cells. So the objects will be, first I'll start with bacteria, resting macrophage, and chemokine and interferon gamma. So chemokine will be TNF alpha and interferon gamma. And I'll try to start with the periodic boundary in the XY. And if I assume the 3D, then in the Z, there will be no flux boundary condition. And we have to give the base value. But the challenge is here to introduce the T cells. So the T cells will come once the number of infected macrophages go above a threshold. So that we can incorporate in different ways. Like if we introduce the T cells after 1000 Monte Carlo step, let's say, and then we will introduce the T cells. So this will be my challenging part here, and then the T cells will move via the chemotaxis. So just up to this part, I'll consider in this hackathon and the formation of early granuloma, and stop the loop after, let's say, 10,000 Monte Carlo steps or something. Yes. So these are the flowchart, but I'm not going into the detail. But that's the thing. These are the resources, and if you want to join me, but I want to write the code myself, but if you want to join me and help me in model building part, it's welcome.
Since there is the existing models from Denise's group that you
Yeah.
mentioned, then
Mhm.
that makes it a lot easier to then do this, because you have a pretty good idea of at
Yeah.
least a potential model that you can use that works to begin with.
Yes. So what I actually thought of is there is a paper that I don't cite here by Elsha Piener. She was also a postdoc
Right.
of Denise's group. So now she's faculty. So
Right.
she has one paper on this agent-based model.
Right.
So I can try to take parameters from her paper. So yeah, that is published recently in M Systems. Yeah. Maybe I forgot to.
Elsa is pretty
Cite her.
accessible, so I'm sure if you asked her, she would be willing to help out.
Mm-hmm. I think, yeah, this one. Yeah, so this one.
Yeah.
Hmm.
Great.
Yeah.
Okay.
Let's see. Yeah, the parameters and the other things I have to, jot down, and then I'll start.
Perfect. Okay. Great.
Mhm.
So we have 2 immune models so far.
Okay. So my goal in general is to develop a model of lymphangiogenesis that I will try to investigate in a chronic inflammation environment. And I try to do it with an experimental model, a microfluidic chip, and then with a computational model. And I try to investigate CompuCell3D to do that. So lymphangiogenesis is the equivalent of angiogenesis, but with lymphatic endothelial cells, so with the lymphatic capillaries. And so the idea of angiogenesis is the creation of new vessels, new branching from an existing capillary or vessel. And so there's a creation capillary. And then upon some kind of stimulation, for example, a growth factor gradient or vascular endothelial growth factor, some cell can differentiate and become, as we call, tip cell that will lead the creation of a new branching like that. And so there are intracellular regulations that make the transition between a quiescent cell and tip cell, and it's how the branching begins. And there were some examples of angiogenesis in the workshop, I think. And so in chronic inflammation, so basically in inflammation, the lymphatic capillaries have a physiological response of enlarging and maybe having a bit of lymphangiogenesis to reduce the inflammation and participate to the immune response. But when the inflammation becomes chronic, for example, in osteoarthritis or in a tumor, we can have the capillaries that are perturbed. For example, we can have regression of the capillaries or abnormal growth of the capillaries being leaky and not working as expected anymore. And so the idea of the project that I have to build an experimental and computational model of lymphangiogenesis is to improve the understanding of the processes involved in lymphangiogenesis. So basically in the tip cell selection mechanisms and the creation of the sprout. And then to investigate, to understand a bit better what is affected to give such abnormal growth or regression of the lymphatic endothelial cells in chronic inflammatory environments. And so the goal of the project is to first have a setup in CompuCell3D that is a bit similar to the setup that I have in the microfluidic chip, so that I can exchange data and information in between the two models to validate or try to test predictions. I'd like to be able, when the model is a bit developed, to test hypotheses on the tip cell selection mechanisms, as we know quite well how it works in blood endothelial cells, but we are still trying to figure out how it translates to the lymphatic endothelial cells. And this could be a platform to try to test hypotheses on the intracellular behavior of the lymphatic endothelial cells and see how it relates in terms of collective behavior of cells. And then I would like to include inflammatory factors impacting the cells and see how, and to investigate the pathological development of the capillaries. And so in the model, I would like to include cell motility, elongation, and the behaviors that could create sprouts, and also adhesion proteins that could be impacted with by inflammatory factors. If possible, to implement philopodia to have the competitive advantage of tip cells to sense growth factors a bit further than the other ones, and then fields of several biomolecules, intracellular rules, and an extracellular matrix. And so here on the bottom left is an example of a simulation I succeeded to get with quite simple rules inside the cells. And on the top left, it's an example of what I can get with the microfluidic chip experiment. If I take only a small height of the channel of the cells, but well, if you are interested in the project, I can explain it a bit more precisely, the model, but there's a collagen gel here and the cell layer on the bottom, and the cells are invading inside the collagen gel.
Like, just want to break in here for 1 second. You've got about 1 minute.
Yeah, sure. Sorry?
You've got about 1 minute left.
Yeah, of course. Yeah, and here's our examples of image analysis that we already try to perform on the images from the experiment, and then we can also apply to the output of CompuCell3D. And so this weekend, I would like maybe to work on introducing ECM in the model, so introduce contact adhesion molecules with the adhesion flex plugin, and maybe investigate philopodia, see if the model could work also in 3D. So I need to figure out on what I will focus really in the weekend, but it's basically going forward with the model. So one reference is the PhD thesis of a PhD from Leiden University that did the thesis on angiogenesis by using CompuCell3D and investigated several ways of model angiogenesis with CompuCell3D. So I'd like to go forward with my project, and if anyone is interested to join and work on it, then you're really welcome to work with me during the weekend. Thank you.
Great. That seems very practical. I particularly like the idea of trying to pair it with the in vitro assays we're doing.
Yeah, it was really the idea to have both models and pair it together to really investigate in several ways the mechanisms.
So next we'll have Nausheen, if you're ready, Nausheen, and then Anastasia, Mark, and then we'll finish things off with Clara.
Hi, everyone. Good morning. Just go through this real quick because I feel like my slides are verbose. I wanted to edit them, but I didn't. So I'm just trying to model, like I do have a previous model of a single cell in Kopazi that sees how hyaluronic acid affects the production of cytokines in the ECM of a CABD patient, like calcific aortic valve disease patient. Just a very, very brief background on what is CABD. It's a progressive disease, and which is like marked by inflammation, calcification, and fibrosis of the aortic valve. And to date, we don't have any cure for the disease other than surgical valve replacement. And it's a very gradual disease, so it's imperative that we find something in the initial stages of the disease. So what I'm trying to do is to build signaling pathways that can lead to the cause of calcification, the calcification and fibrosis process in the valve disease. So what I'm trying to look at right now is how hyaluronan, naturally occurring GAG that is present in the ECM of cardiac tissue, it affects how the disease progress. So what happens is that it's a naturally occurring GAG which provides structural support to the valve, but during the disease process, the ECM is remodeled, and what we have observed in literature, what we find is that the hyaluronan or hyaluronic acid content and organization, it changes during the disease progression, and it directly correlates to fibrosis and nodule formation, calcific nodule formation. So there are two types, basically the hyaluronic acid is basically classified into two different types of hyaluronic acid, depending on their molecular weight. There's a high molecular weight hyaluronic acid and a low molecular weight hyaluronic acid, and they not only differ in size, but also their functionality. The high molecular weight hyaluronic acid, it's a more stable anti-inflammatory a molecule that is present in the normal tissue that promotes tissue integrity and gives stability to the tissue. But what happens during the disease state is that low molecular weight hyaluronic acid is formed, it just breaks into low molecular weight hyaluronic acid, and it becomes less stable and just stimulates some signaling pathways that basically lead to the production of cytokines such as TNF alpha, etc. So this is like a rough outline of the model that I've built in a single cell in CompuCell3D. And what it's just a basic outline of how low molecular weight and high molecular weight hyaluronic acid, they compete to bind to CD44, the receptor, and then kind of initiate a signaling pathway that leads to either the production of TNF alpha, the cytokine, or actually inhibit the production of it in case of high molecular weight hyaluronic acid. So these are the results of my CompuCell3D model. It's a bunch of plots, should have put them in different plots, but what I've found is that the cytokine production increases with the increase in low molecular weight hyaluronic acid, whereas the opposite happens when high molecular weight hyaluronic acid is present. So what I'm trying to do is put this CompuCell3D model in CC3D to see the effect in multi-cell and eventually like at tissue level, what happens and if calcific nodules are being formed or fibrosis is taking place or not. But that's like a bigger goal. But for the weekend, I just want to implement somewhat some of the model and like just see some of the results and just to see how CC3D will work with my model. So I do think that this is a feasible project that can be done in CC3D. Just to see the changes in different types of cells. So I will have like two different types of cells, the endothelial and interstitial cells in the model with the field cytokine in it. And I just want to see the output if these cytokines will lead towards calcification or not. So I will try to work on the model on my own, but if anyone is interested in contributing towards working as a team, I would be more than happy to welcome any teammates. That's it from me.
That's great. I think it's important to begin to think about what extracellular and cellular components you're going to need, and also the spatial organization of the tissue you want to model.
Yeah, I do have a colleague that worked with the TGF-beta pathway previously. His paper came out last year. So I'm gonna just try to like do the spatial modeling from his paper. It won't be exactly the same, but it would be similar to that.
So I mean, you'll need at least the high molecular weight and low molecular weight hyaluronic acid fields, possibly.
Oh, okay.
I don't know. You have to think about that. But
I'll have to think about it.
It's a great problem, and you have the intracellular model already. So one thing you can try to do then would be to replicate, as you begin to build a multicellular model, you have the time series data from the ODE model that you already have. You could see whether you're able to replicate that, for example. So those are all possible.
Hi, thank you, everyone. This has been really just a completely wonderful experience, and I've loved all of it. I already presented my project, so very briefly, it concerns the motility of neutrophils in mucus under different conditions. So the motivation of this is that neutrophils must traverse mucus in order to capture and eliminate pathogens. So this is a sort of extracellular environment that they always have to deal with, but we don't often think or talk about. And there is experimental data from multiple sort of different methodologies and systems that shows that non-physiological mucus can impair neutrophil motility. So, for example, in disease stage such as cystic fibrosis, the mucus becomes very thick, and then that can result in reduced motility of neutrophils. So the key question for this project would be, how do disease-associated changes in mucus properties affect neutrophil motility in 3D? And the objectives would be to help us understand the immune defense in mucosal tissues, and then potentially reveal mechanisms of disease progression. That's sort of the purposes of asking that question. And then more specifically in terms of the modeling approach, it is very impractical to test all possible mucus conditions in vitro. There are in vitro experiments that would be great to conduct, but a model would help us prioritize the regions of the parameter space in terms of the mucus properties and mucus pathology that we can explore when we're actually testing things out in vitro or even later in vivo. So the goal of this project would be to generate sort of a phase diagram where we're mapping neutrophil migration behaviors across different mucus conditions. And then the applications of that most directly would be to inform in vitro experiments. So we basically figure out what concentrations or what porosity or what perturbations in mucus might give us interesting phenotypes that are relevant to disease states and that can also inform in vitro experiments to do. And then most ambitiously, if we can have such a model, it might be able to allow us to map, if we have information about the mucus in a certain disease or even in patient-derived mucus, to sort of predict disease progression based on neutrophil infiltration. And then we can also use that to sort of simulate potential therapeutic strategies. For example, if we engineer neutrophils to do X, Y, Z, how would that affect their ability to infiltrate diseased mucus environments? And yeah, based on what we've learned so far, it seems like a very feasible project to at least get started on, to at least have the basic setup on, because we've talked about chemotactic cell migration, and there already is a CompuCell3D model on mucosal barriers that we could use as a starting off point. So these are the citations, and I want to draw your attention to the images. The one on the left is the model that has already been published. I was looking at the interaction between mucus and bacteria. Then the schematic in the middle is the in vitro experiment that I think would be great to begin by starting to reproduce. So basically you have the neutrophils at the bottom, and they have to traverse this mucus layer to reach the chemotactic signal at the top. And so this was an actual empirical data that is showing that in thickened mucus, you get slower neutrophils, and the infiltration of the neutrophils through the mucus layer is hindered. So I think a good starting point would be to try to make a model that can reproduce that result. And then if we have set that up, we can see how we can extend it or explore different parameters. Yes, I have a PhD in molecular developmental biology, but this is related to what will very likely be my postdoc project. I haven't started yet, so I need to learn. I'll need to be reading up on this along with everyone that might join. And I already have a teammate, Joanna, who's a PhD student in molecular biology. I'm very excited to work with her. And if anyone else is interested in the project, I think we'd be happy to have 1 or 2 more people join. Doesn't have to be any sort of specific skills that you have. If you're interested in this, if you think that any part of this is something that you might be interested in modeling, I think that'd be great to have you on the team. Thank you.
Okay, so I think one key thing, and it sounds like you've already started thinking about it, is how you're going to represent the ECM, how you're going to represent the mucus. Is it going to be material made up of blobs of material? Is it going to be a field? Is it going to be a vector field? Is it how are you going to represent this complex extracellular material? And there may be many different ways of doing that, but for the weekend, it's a question of how you start with that. So I think that may be the most challenging part of this project. And if you've got existing models that have been published to do that, that's a great place to start.
So my name is Mark, and my hackathon project it's an extension of what was my master project that I finished the last month, and it's on vegetal rotation in explants. And so in my master project, I was studying gastrulation in Xenopus laevis and how the first stages of development, where we go from one egg to through gastrulation and then neurulation, and the organism is much more developed. And so actually, if we cut this egg and we look to the sagittal section, what we observe is that, there is huge movement of tissues inside of the embryo. And so actually during gastrulation, which is a process I focus, there is the internalization of the endoderm and mesoderm, which is crucial for the development of the organism. And so during my master project, what I was trying to do was the gastrulation from stage 10 to stage 10.5 using cell pods model and doing the simulation with 5 different tissues and different contact energies and so on. However, there is this paper by Jason Wan and Rudolf Winkelbauer when he basically looked into what happens when they cut explants from embryos. And so one of the particular things that is interesting in gastrulation is how the endoderm is internalized. And this is done through vegetal rotation. And this occurs by a change in the overall shape of this tissue. And so when they cut these embryos and they had these explants, what they saw was that actually when they were outside of the embryo, they were still observing the vegetal rotation, this movement of the cells. I put it again. And so my project is to try to make a simulation of this with CompuCell. And so the idea would be to start using cell ingredients on a bottom-up approach. This is, we would start with only volume, surface, contact energy plugins. And then, of course, if there is migration, we can try to explore different ways to achieve this migration. And also it's a bit challenging because we want a collective migration because there is an overall change of shape. And so one of the things that they describe is that there is an amoeboid migration. So maybe we can use compartments to describe cells. And also this problem is 2D, as I presented, but it's also true that there are some cells that are appearing from different planes. And so we could extend it to 3D. And finally, I would also be very interested to make plots on different kind of measurements that we can do on videos taken from microscopes, but for CompuCell. And so this is also part that would be interesting to include in the project. I think this project could be feasible because I have already started my project using CompuCell, and it has looked quite good so far. And so the input would be to use 1 or 2 tissues at most with very simple plugins, and then the ideal output would be to understand how vegetal rotation is still working without any adjacent tissue. And so it's occurring inside the same tissue. And also to have some tissue properties, measurements that could be recycled for your future projects. So if we manage to measure, thank you, Hayden. If we manage to make measurements of pressure, of tension, of velocity, all of these could be recycled in the future probably. And so if this works, then I would include this in my current simulations with the 5 tissues. And we would see how much do we win with this detail in the endoderm. And so the outline for the weekend will be to explore different migration strategies, to explore different levels of cell description, and prepare different plots to analyze the simulation. These are some of the resources. This is the main paper, but there are some others. And my background is I graduated in math and physics, and I have some experience in molecular dynamics, and for the last 4 months in morphogenesis. But for this project, I will be very happy to welcome anyone, either biologists who want to start in modeling, or people who has already some experience in modeling but want to apply some of the things that we've seen in this workshop, and it will be also interesting. And thank you for your attention.
That's great. I'm looking forward to seeing what you've already gotten working there. I worked with a student who for a while who was working on a somewhat similar project in the explant reorganization of tissue. So I'd love to see this happen. That work by Job on neural tube closure may be helpful because he was able to get that hinging movement of the neural tube on a large scale, and so the ways he implemented that might be things that would be helpful here too.
So my name is Luis. I'm from Meiji University in Japan. Thank you for letting me share my ideas with everybody. I hope someone can join to my work. We are trying to develop a dynamic 3D scaffold for cell behavior control using SALSA, what is called as a scaffold simulator, and CompuCell3D. So the okay. The biological background, I mean, I'm not a biologist, but anyway, our main idea is that the tissue engineering requires some scaffold to mimic the dynamic in vivo environment. So we focus on the shape memory polymer, which is in the right side. It's just an idea of how it should behave, is that it deforms in response to the temperature. For example, the temperature between 20 and 40, there are several shape memory polymers. But for example, all polymers have a critical temperature, which above this critical temperature, the polymer will try to recover their original shapes in the right side. So we are trying to use this movement of the polymer and let cells inside these holes or this space move inside this space. And based on the movement of the polymer, we can try to study or try to have some idea about how we could control or how we can modify the behavior of these cells by changing the environment with the temperature control. So the problem, my main motivation was that the dynamic of the this mechanical movement of the scaffold, I mean, contraction or expansion, could modulate the cell behavior. So this it will be let us control how the cell move or improve also the tissue engineering or maybe some personalized implant. So we'll try to understand also how cells can react to scaffold deformation to allow this smarter scaffold with time-dependent clips. So, and the modeling goals will be to try to simulate this scaffold deformation with under some heating using a thermomechanical model. This is a point for discussion. Maybe we don't need to use this model, but maybe we can include everything without CompuCell3D. But anyway, we would like to simulate this cell migration and adhesion inside this scaffold using CompuCell3D. And maybe we can link both simulator to see the dynamic deformation, if this dynamic deformation alters some cell distribution. So we want to predict where the scaffold movement can see some cells to move this cell to specific locations and something like that. So the feasibility of the project, well, is CompuCell, we can combine this both. Thank you, Hayden. So combine with two simulators or not. And let's say the input will be the scaffold geometry, the 3D geometry of the scaffold, the thermal properties, and then some migration and positions related with the cell. And what we want to achieve is the scaffold shape, how it changed over time, and what happened with the cell location, density maps, what adjacent zones for these cells. The steps will be to build the scaffold, simulate and export the frames for the scaffold and convert this mesh to 3D, and how to simulate this visualization. So the results, there is some page for this scaffold, was this both simulations with their papers, respectively papers. And I would like to, so I've been working in biomedical and mechanical engineering. I have some background mainly from machine learning and computer vision and human-computer interaction. But I would like to join some, or I would like to continue learning as it was made in the workshop from the interaction with my possible tentative teammates by doing CompuCell scripting or simulation workflow and try to collaborate with more biologists or cell behavior modeling in my talk. Thank you very much.
So just to be, you didn't show us what salsa does. So maybe next time you can show us some pictures of how salsa works.
Yes.
Because, and so the other question I would have is, do the cells actually remodel the ECM, or is the ECM purely something the cells crawl on? Do the cells actually change the ECM, or do they only respond to the ECM?
Actually,
the scaffold.
I don't have the, how to say, for my polymer, I don't have the how to say, the experiment with the polymer yet. So my focus is try to make simulations to know what will happen with the experiment later, what could happen. But as I know, I don't know, the biologist background maybe could answer better than me. The cells can receive the nutrients through the scaffold, and they can consume or degrade the scaffold also, I guess. So I'm not familiar with this part, but with my design, I would like to both ways. So the cells could affect also the scaffold, and the scaffold could move the cells. But the movement of the cell could be a feedback for the scaffold movement itself. I don't know if it answered your question.
Well, you're going to have to make to build a simulation. You're going to have some hypothesis about that. So you can change your hypotheses or have multiple ones, but you're going to have to decide, what the scaffold does to the cells and what the cells do to the scaffold, because that's going to be a totally separate problem from how the scaffold changes size and porosity when you change the environment. So I think trying to subdivide your problem into pieces, smaller questions, may make it easier to solve. The big problem is fascinating, but now the question is, could you define sub-questions that are a little more manageable to start with?
Hello, everyone. My name is Harini Raghukumar, and I am a graduate student at Michigan State University. Me and my team, Aryan, Aishwarya, we are all Aurora Lab members. My PI is also here during hackathon. She was also here for the entire workshop. So here we are aiming to model mesenchymal regulation of epithelial folding in a pregnant uterus. It is known that the epithelial folds exist in female reproductive organ, but its role in pregnancy is something that's not known. And it is established that about 70% of the conception 70% of the conception is lost prior to birth, and about 30% of the conception is lost around pre-implantation stage. So we believe that epithelial fold defects could contribute to these losses, and therefore we are interested in knowing how folds are formed in the uterus and how they might impact pregnancy success. So we use mouse model to understand epithelial folding in the uterus. The mouse has two uterine horns. If you take a uterine horn, the center is the lumen made of epithelial cells. The mouse uterus is a multi-layered organ containing the smooth muscle, the mesenchymal layer, and the luminal epithelium. So to visualize the organ in 3D, our lab has developed a technique to clean, stain, and 3D image to analyze all these layers. From our previous work, we know based on 3D analysis that the lumen we have identified multiple folding patterns. So I'm just quickly going to explain the different folding patterns with the help of a pregnancy timeline in mice. The time of mating is considered as gestational day 0, and we visualize a plug at gestational day 0.5, which is the morning of that day. Once the pregnancy is successful, the pups will be born on day 18 or 19. So if you look at the schematic representation, you can see that the horizontal folds we have defined it as longitudinal folds, and the vertical folds here are defined as transverse fold. On day 2 of pregnancy, we observe more of these horizontal folds. And as we move 12 hours further, that is on day 3 of pregnancy, we see all the folds to be horizontal like this. And this is how our 3D images look like. Once the embryo enters and scatters in the uterus, which is 6 hours from this time point, we see all the folds to change to these vertical patterns. So this is how it looks like in 3D model. The reason why we want to do simulation using CompuCell is because we know that all of these happens, and these are all snapshots of different time points. We cannot really do live imaging on the uterus because we are technically having some issues with culturing the uterus. So we believe that simulation of our model might help us understand how these folding pattern changes and what's the reason behind it, how the folding changes. So our aim here during the hackathon is to focus especially on this region. Our first goal is to make a single layer of epithelium and then the ECM, then 3 layers of the mesenchyme, then make a fold like this, like you see in this drawing, and then see how the folds change by adding the external forces, and then what happens with by adding mitosis or migration in the mesenchymal layer. Priyam's talk on Tuesday was very helpful for us, and we are thinking of building from that model that she was explaining the other day. And as of now, anyone with expertise in coding or modeling or any mathematical background are welcome to join the team. Thank you.
Okay, can you remind me of the scale here in this image? What would the scale be that we're talking about in this box that you draw?
Oh, so I didn't have time to draw individual cells, so I just made one layer of epithelium and one layer of ECM like that.
But you're talking about
how many microns? What is the fold? What microns? Is that what you're asking?
right. But it's how big a domain do we have to simulate?
Look at your cryo sections, Harini. How deep is the fold? That's what they're asking. I think it's about 3 or 4 cell layers. So that would be about 50 to 60 microns between, yeah.
Well, certainly if you can start with something like that epithelial folding model that Priyam showed, that gives you a place to start that you could work from. Very interesting.